We're going to continue our discussion of the nervous system by discussing the cells that are called neuroglia. Neuroglia are cells in the central nervous system that support the neurons. There's one neuroglia type cell that is outside the central nervous system. We've already talked about it. It's called a Schwann cell, and it is responsible for producing the myelin sheath in the peripheral nerves. The first of the neuroglia that I want to mention is called an oligodendrocyte. An oligodendrocyte is a supportive cell in the central nervous system that has two functions. It is the Schwann cell of the central nervous system in that it forms the myelin sheath for neurons in the central nervous system, and it is also responsible for helping hold together neurons in the central nervous system. The second of these neuroglial cells is called an astrocyte. And you may have heard of this before because brain tumors, the most common brain tumors, are called astrocytomas. And the most common of those is called a glioblastoma multiforme, or a grade 4 astrocytoma. Anyway, this is another supporting cell in the central nervous system, and it's responsible for producing something called the blood-brain barrier. There is actually a barrier or a filter that filters between the blood and the brain so that materials do not directly transfer from the blood to the brain and back and forth. Some things are able to penetrate the blood-brain barrier more vigorously than other things, but this barrier is there to try to protect the brain from things that might be in the blood that might not be good for the brain. The third of these neuroglial cells is called the microglia. Microglia are the infection-fighting cells in the central nervous system. They help fight infections. They engage in phagocytosis, so they eat foreign material and microscopic organisms, and they help fight infections. Now, another one of the neuroglial cells is shown in this diagram, and it's seen all along the side there on the left, and these are called ependymal cells. Ependymal cells are cells that secrete what's called cerebral spinal fluid, and it also, because of the villi on these ependymal cells, that circulates the cerebral spinal fluid. Now, cerebral spinal fluid is fluid that surrounds the brain and is inside the ventricles of the brain, and also travels up and down the spinal cord and acts as a cushioning area for the brain. The not shown in this diagram is something called a association neuron. And I mentioned this in a previous slide, that an association neuron is a neuron that transmits action potentials from one neuron to another inside the central nervous system. Now, when we look at the peripheral nerves, they have a particular structure, which is shown here, and I'm going to go through the various numbers here and tell you what each of these are. The first is the outer sheath that's around the outside of the neuron. It's a very thin structure, and it's called the epineurium. Then inside of that is the actual nerve sheath, and this binds together these bundles of axons. Now, the bundle of axons is called a fascicle. So each of these little sections or bundles is called a fascicle, and it represents several axons bundled together. And the bundle of axons are held together by something called the perineurium. Number 5 here is the actual covering around the axon. It's called an epineurium. And then the 6 is the actual axon itself. And so you have a bundle of bundles, which is surrounded by the epineurium. Each individual bundle is surrounded by the perineurium, and then each individual axon is surrounded by the endoneurium. And notice the name, epi for outside, peri for in between, and endo for inside. And that's one way it helps you remember. You'll notice something when we get to the muscles. It's going to be called epimysium, perimysium, and endomysium. The same sort of structures. So in this, though, we have the epineurium surrounding all of the fascicles. We have a fascicle surrounded by the perineurium, and then we have a axon surrounded by the endoneurium. And then in between are fatty structures, and there's also some blood vessels that you can see cut on end in here. Now, you remember that we divided our neurons into motor and sensory, and the motor neurons and the motor neuron here carries the impulse from the central nervous system or the spinal cord to the muscles and activates movement. And it's actually shown here as B. You'll notice the little arrow shows that the impulse is going toward the muscle itself. The sensory neuron carries the impulses to the central nervous system, and this is shown as A. And in this case, it's sensing maybe that the muscle is being lengthened. This is also a picture of a reflex arc. And what happens in a reflex arc is the muscle is stretched first. That impulse is carried on A. Then in the spinal cord, a impulse comes out on B and causes the muscle to twitch or contract. And then nerves that carry both types of impulses are called mixed nerves. Now you'll notice that I'm talking about nerves now, not neurons. Neurons are committed one way or another. They're either sensory, motor, or association. Here, nerves can contain all motor neurons, they can contain all sensory neurons, or they can contain both sensory and motor neurons, and they can carry both types of impulses. Now we're going to spend the next several minutes talking about how nerves conduct their impulses. And the primary way in which nerves conduct their impulses is something known as the action potential. And the action potential is an electrical phenomenon. And in order to understand this, you have to understand how the electrical situation is set up. And I'm going to spend several slides trying to explain this in several different ways to help you understand it. So the action potential is the impulse that's carried down the neuron. And this impulse travels down a neuron axon, begins in the axon hillock, and travels down the axon to the presynaptic terminal. And that's the direction that it goes. In order to do this, the cell has to be excitable or have excitability. And that's what this term excitability means. It means that it has the ability to undergo an action potential in response to a stimulus to do that. And the basis of an action potential is the fact that there is a difference in charge, electrical charge, on both sides of the membrane. So this difference in electrical charge, comparing the outside of the neuron cell membrane to the inside of the neuron cell membrane, is called the potential difference. And the potential difference, and I'm going to go through this diagram briefly, and then, like I said, we'll go through it in several different ways to try to help you understand how this happens. So let's look first at number one here. Number one is the resting state. Now in number one, you'll notice there's a couple of things that look like coffee beans and another one that looks a little weird. The yellow looking coffee bean is what's called the sodium channel. The more orangey coffee bean is what's called the potassium channel. And then this green, unusual looking thing is called the sodium-potassium pump. And the only thing that's working when we're at rest is the sodium-potassium pump. And the sodium-potassium pump pumps sodium out of the cell and keeps potassium in the cell. And you'll notice in picture 1, there's more sodium on the outside than there is potassium on the inside. So that means there's more positive charges on the outside than positive charges on the inside. And therefore, if you look at the graph, number 1, the potential is around minus 70. The y-axis of this graph is the membrane potential in volts, millivolts, and the x-axis is in time, in milliseconds. And so all this happens in a period of time. So number 1, the 2 channels are closed, but the pump is working to keep the potential minus 70. Now you know that the natural inclination, if everything was passive, the natural inclination would be to approach 0. So this pump has to work to keep it at minus 70, and it uses energy to do that. Now when the action potential goes forward, the first thing that happens is that sodium pump opens up. And if the sodium pump opens up, by diffusion, sodium rushes into the cell. When sodium rushes into the cell, the inside of the cell becomes more positive than the outside of the cell. And then you see that the membrane potential changes and approaches 0 and actually goes a little bit over 0. And this process is known as depolarization. Then, the third part, the sodium channel shuts and the potassium channel opens and the potassium rushes out. And this tends to reverse what just happened with the sodium and causes the potential to go back down into the negative territory. And then at some point, number 4, you reach the resting point again. Both channels then close, and the resting potential is then established again by the pump. And this process, over a period of about 5 milliseconds, is the propagation of it. This happens in one section of the membrane, and then the next section of the membrane, and then the next section of the membrane. So along the length of the membrane, this potential is traveling down from the cell body toward the presynaptic terminal. And that's how it conducts the action potential down the axon. So let's look at it just in another way. We said that the difference across the membrane, the extracellular fluid is on the top here, intracellular is on the bottom. The difference in charge is across this membrane is called the resting potential when there's not an impulse. This corresponds to number 1 on our previous slide. There's this pump that keeps this happening. You can see here there's a couple of different diagrams of there's 3 of these pumps here, and each time an exchange occurs, a sodium is pumped out of the cell, potassium is pumped into the cell, and you'll notice there's ATP associated here. And the ATP, the purpose of the ATP is to provide the energy necessary, because this is against natural laws. So you have to use energy to go against natural laws. If you start on the left, you'll notice that the sodium is collected and the ATP is bound. The energy, at the same time, potassium is collected on the outside. ATP is bound, and this pump then turns and pumps out the sodium and pumps in the potassium. Here's another diagram showing basically the same thing, but it has a couple of nuances that I want to point out. First of all, the action potential is what is called an all or nothing event. This means that the event either occurs at its maximum effectiveness or it doesn't occur at all. There's no such thing as a partial action potential. It either happens or it doesn't happen, and that's all there is to it. And as I said before, the initial event, when the sodium rushes into the cell, there's a loss of the difference in action potential, or the potential difference. This loss of potential difference across the neuron cell membrane is called depolarization. And this is shown on the diagram on the left part of this little hill here. So here in just review, we start out more sodium on the left side here. We have more sodium on the outside and potassium on the inside. There's more negative charge on the inside than on the outside. So this is the first point. Then with depolarization, sodium rushes in, and that causes the potential difference to go towards zero and actually go above zero to about 50. Then potassium then rushes out, and this represents the repolarization process. And then the last part is what's called long-term active transport, which just means resting state, or it's part of the resting state.